Hello everyone, my name is Ibrahim Khalil from Group 16 and these are my friends, Haslinda, Yogesh and Najwa. Today we are going to talk about the biosynthesis and metabolism of purines and pyrimidines. Buckle up because we have a lot of stuff to cover.
Okay, for the introduction part, purines and pyrimidines are two classes of nucleotides in the cells. There are two types of purine which are adenine and guanine, while cytosine, thymine and uracil are the three types of pyrimidines. Nucleotides also serve different functions in the cells such as building block of DNA and RNA, energy carrier, component of coenzymes, and also second messenger in signal transduction. Next, metabolism of purines and pyrimidines has three different processes. The first one is de novo synthesis, followed by degradation or also known as catabolism, and the last one is salvage.
De novo purine synthesis is the process of producing purine from scratch. The de novo purine pathway starts with the synthesis of IMP, then branch into two paths of either synthesizing AMP or GMP. The branch point is controlled by feedback inhibition, where the AMP would inhibit the conversion of IMP to GMP and vice versa. Next, let's look at the overview pathway. First, ribose 5-phosphate is converted into PRPP by PRPP synthetase in a step requiring 1 ATP. Then, in the committed step in the process, an alpha amino group is then added to PRPP from glutamine to form 5-phosphoribosylamine. This reaction is catalyzed by glutamine PRPP amidinotransferase. After that, a series of 9 reactions result in the formation of IMP. Lastly, IMP can be transformed either to GMP by IMP dehydrogenase or to AMP by adenylosuccinate synthetase. Purine catabolism pathway. Purine catabolism pathway is the breakdown process for purine. The catabolism pathway starts with the breakdown of the nucleotides to its respective bases. The bases will then be further broken down into uric acid to be excreted. For GMP are hydrolyzed with nucleotidase to form guanosine. The guanosine then undergoes phosphorylases with nucleoside phosphorylase to form guanine. Next, for AMP, usually AMP are first deaminated by deanylate deaminase to form IMP again. The IMP then undergoes hydrolysis with nucleotidase to form inosine. Sometimes adenosine can be present and is deaminated by adenosine deaminase to form inosine. Next, the inosine undergoes phosphorylasis with nucleoside phosphorylase to form hypoxanthine. Lastly, for base, guanine are deaminated to form xanthine. Hypoxanthine, on the other hand, is oxidized by xanthine oxidase to also form xanthine. The xanthine then undergoes oxidation with xanthine oxidase to produce urates and hydrogen peroxide.
Okay, for the purine salvage pathway, it is defined as the recycling of purines formed by degradation of nucleotides. In comparison to de novo pathway, salvage pathway is energy saving. Purine synthesis occurs through salvage pathway in all tissues. This is extremely significant in the brain and bone marrow. Substrate involved is hypoxanthine, PRPP, guanine and adenine, while the product form is GMP, AMP and IMP. Okay, for the pathway overview, APRT converts adenine and PRPP to form AMP. Next, hypoxanthine can be combined with PRPP to form IMP in a reaction catalyzed by HGPRT. Subsequently, IMP can be transformed into AMP or GMP through the last few steps of the de novo purine synthesis pathway. HGPRT also catalyzes a reaction that combines PRPP with guanine to form a GMP.
Synthesis pyrimidines de novo. The making of pyrimidines nucleotides initiating from the cell's building blocks forming the biochemical pathway is called as the de novo pyrimidine synthesis. This de novo synthesis for pyrimidine process starts up with the synthesis of ring forming pyrimidine nucleotide after the attachment to the ribose phosphate. The cytosol of the cells found in all the tissues is where the de novo synthesis occurs. The process of this pathway is initiated with the combination of both carbon dioxide and glutamine, producing the carbonyl phosphate, where the reaction is catalyzed by a catalysis called carbonyl phosphate synthetase 2. When the carbonyl phosphate is mixed with aspartate, that might be subsequently dehydrogenated and water to form the orotic acid through a series of steps that would be inhibited by leflunomide. After that, orotate phosphoribosyl transferase catalyzes the orotic acid that is attached to the ribose 5-phosphate. This reaction has an end product of orotidine 5-monophosphate. The OMP is decarboxylated to form a major pyrimidine nucleotide that is a precursor to RNA called uridylate by OMP decarboxylase. Then UMP is phosphorylated to form UTP. Subsequently, the UTP is converted to CTP with the addition of an amino group from the synthetase. This conversion of UTP to CTP is catalyzed by CTP synthetase. The pyrimidine catabolic pathway is thought to be also involved in the degradation of pyrimidine-based antimicrobials. The products of pyrimidine degradation are more water-soluble than other products of purine degradation. The pyrimidine bases in most organisms are subjected to reduction rather than oxidation. Pyrimidines can be degraded reductively, oxidative by attack of oxygen in the pyrimidine ring, by oxidative demethylation of thymine, by decarboxylation of orotic acid to uracil, and by reductive degradation of orotic acid. Pyrimidines are ultimately catabolized to carbon dioxide, water, and urea. First of all, cytosine can be broken down to uracil, which can be further broken down to N-carbonyl beta-alanine, and then eventually to beta-alanine, carbon dioxide, and ammonia by beta-uridopropionase. Whereas the thymine is broken down into beta-aminoisobutyrate, which can be further broken down into intermediates, eventually leading into the citric acid cycle. Beta-aminoisobutyrate acts as a rough indicator for the rate of DNA turnover.
Salvage pyrimidines is nucleosides and free bases generated by DNA and RNA breakdown are converted back to nucleotide monophosphates, allowing them to re-enter the pathways of pyrimidine biosynthesis. Nucleotide conjugated sugars act in cellulose biosynthesis and are involved in glycosylation and lipid metabolism. Because the biosynthesis pathway of a de novo pyrimidine consumes energy, plant cells reuse pyrimidines base and nucleosides derived from performed nucleotides as in figure. Basically, only result is reused directly through certain phosphoribosyl transferase. Rhodine, cytidine, and deoxycytidine are exclusively salvaged to their respective nucleotides. For summary, research on nucleotide biosynthesis and metabolism in plants has provided a framework for more detailed analysis of specific enzymes and the isolation of their corresponding genes. Availability of a complete Arabidopsis genome sequence will aid in the search for the encoding sequences of purine and pyrimidine biosynthesis enzyme and catabolism based on their similarity to sequences. Housekeeping enzymes, such as those involved in nucleotide metabolism, are often thought to be expressed at a consistent level in all cells. However, it is now proven that genetically expressed activities are constitutionally highly dynamic so as to meet the demands of plant cell metabolism. Since these changes can have a major impact on crop development, there is a new appreciation for the importance of housekeeping activities in plant survival, such as in a response to stress.
Well, that is all that we have to say for now. I hope you guys have fun watching this video. Looks like it is time for us to go. We wish to see you guys soon. Bye bye!